of introduction about the Institute. We are a collaborative research institute for theoretical computer science, as well as its connections to all other areas of sciences, engineering, humanities, and whatnot. And we run a pretty packed schedule. We have typically 2 programs each semester. And also a few years ago, we started summer programs, which is, is one of them. And each program has, between, close to 50 to 70 long-term visitors, both junior and senior, who come here for workshops, collaborations, and so on. And this semester, the summer term, we have many programs, of which the computational innovation and data-driven biology is one of those. And special thanks to the Correct Foundation for its support of this program. And welcome to the first workshop in this program, from algorithms to discovery in genome-scale biology and medicine. So first, I want to thank, on behalf of the Simons Institute, the organizers, Nada Vahaytu, Rodet Charan, Stephen Brenner, Ron Shamir, Yun Song, and Olga Troyanskaya for arranging an excellent set of speakers for this week. And I'll hand over to the organizers for introduction to the workshop itself. So here, I'm just here to welcome you and give some logistical information. So just some logistical information, snacks and drinks and coffee, etc. will all be outside. And for lunches, you're on your own. And please enjoy your food and coffee outside. We do not allow any food or coffee in the drinks in the auditorium. Water is from your water bottles are okay, of course. And the policy right now is masks are optional, but we strongly recommend that when indoors, you mask up if you can. And there are lockers just outside where if you want to store your backpacks during the day, you can, and they're very easy to use. If you want to use Wi-Fi, EduRoam works. You can also sign up for Cal Visitor. Tends to be a little more flaky, but if EduRoam works from your home institution, that's best. And I was just told we are not using a hybrid format, so this will be an all live, good old style workshop. So good luck with that. And that's it. And finally, I just want to thank Kelani Penland, who's standing outside there, our events coordinator, for all our work on the local arrangements to make this event run smoothly. And if you have any questions, she'll be around. Feel free to find her and ask her. Okay, with that, welcome again, and let me turn it over to the organizers.
Hello, I'm Ron Shamir from Tel Aviv University, and together with Stephen Brenner from UC Berkeley, we are the co-PI of the Correct Berkeley Tel Aviv initiative in computational biology and bioinformatics. So this is an ongoing initiative that is in its 4th year by now, and it fosters collaborations between Tel Aviv and Berkeley in the field of computational biology, and it's funded by the Correct Foundation. Finally, after 3 tries, we are having this summer program. It was planned for 2020 and again for 2021, and finally we could make it happen now. So we are going to have 2 conferences. This is the first one, and there are also about between 2 and 3 dozen students and PIs who will be here for all or most of the 5 weeks of the program for interaction and collaboration. So I think the persistent heads of the program for doing it again and again until it actually happened, and the team at the Simons for all the help and support. Stephen probably wants to say a few things.
So I just want to welcome everyone here to Berkeley. It's great to have you here. As Ron said, I remember back in 2018, planning in 2019, then in Tel Aviv, planning out details of this meeting and then planning them again and again and again. I think it's perhaps appropriate that within the last week, we've had to change our first speaker twice for this meeting, but it's great to see all of you here, physically here, and have this meeting happening. Nadav and Roded really did the greatest lifting here and making this happen. We're really grateful to them for doing this over and over and over again. I think that we have a great week of science ahead. And so I want to thank them. I want to thank all the speakers, all of you who joined us. And again, also, we are really appreciative to the Court Foundation, which has sponsored this incredible program that's allowed us to have the collaborations between Berkeley and Tel Aviv University. If anyone has any more questions about that program, you can look at our website or ask me or Ron any more questions. So thank you very much and welcome.
So hi, I'm Oded Charan from Tel Aviv University, and I think as Ron and Steven mentioned, it's quite a miracle that everyone is here. Not everyone, actually my co-organizer, Nadav Achituv, couldn't make it because of these hard times, but he might join us tomorrow, at least for the brainstorm session online. So let me start from the end. I really wish to thank all of you who made the effort and managed to come here despite everything, despite COVID, despite the airports, despite whatever. So I'm really grateful for all of you. Okay, so this workshop that we are starting, which is called From Algorithms to Discovery in Genome-Scale Biology and in medicine, will include cutting-edge talks in genomics and systems biology, which will cover both algorithmic development as well as biotechnological innovations. And more specifically, today we will hear talks on gene regulation, highlighting the importance of spatial omics or spatial omic data. And then we will have the poster session as well as the reception. Tomorrow we will hear talks on precision medicine. And tomorrow we are also planning this brainstorming session on the future of the field. And I really urge you, anyone who hasn't yet completed the questionnaire that we shared with you, please input there your feedback. On Thursday, we will hear algorithmic and technological development talks in network biology. And finally, on Friday, we will focus on the problem of predicting phenotypes from genotypes. And let me just wish all of us success in this meeting. Thank you. Yeah, so our first speaker is Shyam. Please go ahead.
Thank you, Roded. All right, I guess it's working. There we go. Right, huge pleasure to be here. Really want to thank Roded and Nadav and all the organizers for bringing me here. I've lived in the Bay Area for 15 years and been visiting Berkeley since 1992. Did a postdoc up the hill. I don't know which way that is. Lawrence Berkeley Lab. Okay, that way. Ron is pointing that way. And also did some experiments at Lawrence Berkeley Lab as a physicist during my PhD. But then I switched to genomics for my postdoc, again at Lawrence Berkeley Lab. But yeah, and so know the space very well, know Nadav very well. Sadly, he's not here. He and I did a postdoc together at Lawrence Berkeley Lab. Spent many years, and I've heard Roded talk in Berkeley. I've probably heard you talk also, Ron, over there, and maybe many of the people here. So it's kind of like coming back home to be here. All right, going to be talking today about spatial omics algorithm that we developed recently and very excited about. And with this audience, I'm really looking forward to some feedback. I think this is the first time I'm presenting it to a proper computational biology audience. So excited about that. So evolution of genomics, high throughput genomics. So we started with bulk genomics. I presume most of you know is just mashed up tissues. And then you count the molecules in those mashed up tissues, which is much easier because if you take a lot of tissue and mash it up, you got millions and billions of molecules to count. And if you're measuring anything, it's easier to measure large quantities than to measure small quantities, right? Your measurements are just more accurate, more sensitive. So that was easy, and that's why people did bulk genomics, not because they wanted to do bulk genomics. It was the only choice they had. Technologically, single-cell genomics was not possible on a high throughput scale. But then, of course, in 2009, I think, was the first single-cell RNA-seq paper. And even before that, I think most people don't know there was some single-cell microarray gene expression papers also. So, and that really changed the game. Now, the beauty of single-cell analysis is that you're no longer mashing up the tissue, right, into this homogeneous mush. You're taking apart the tissue into its component parts and looking at each part individually. And then you get a much better idea of what's happening in that tissue, right? But obviously, you know, this analogy, you know, there's one more image here, there's some empty space here. This is incomplete, right? Because you want to know how these things fit together. You really want to see this, right? You want to see the component parts of the tissue in their spatial environment, in their spatial architecture. Then you really understand their function and interactions. And that's what was missing, even though single-cell technology was incredible and powerful, and it's kind of becoming the default for expression profiling in tissues. What you really want is spatial genomics, all the power of single-cell, measure all the genes, all the expression, chromatin, everything, without destroying the tissue. Just keep the cells where they are in the tissue. Right? That's the dream. And now there's an explosion of spatial omics technologies, lots of startups, lots of big companies getting into that space. We are collaborating with one of them for the technology development. And so not surprisingly, right? So in 2020, Nature Methods named Spatial Omics as the method of the year. And before that, single-cell RNA-seq twice won method of the year in 2013 and 2019. So what is spatial omics? So you get data like this, okay? This is the dentate gyrus of the hippocampus. I think it's the mouse brain hippocampus. Each little blob or dot, each blob is a cell. And you can see here there are different colors. I think that either the cells are colored by cell type or they're coloring molecular markers of those individual cell types, right? So here you can see beautifully all the richness and cell type diversity of a complex tissue and its spatial architecture, right? This is what you want. You want to be able to measure all, let's say 20,000 genes or 30,000 genes, whatever, measure their expression in the spatially resolved way. It's not really possible right now, or it's possible in principle, but at low sensitivity and often at low spatial resolution. Ultimately, you want subcellular molecular resolution, single molecule resolution, and you want to be able to have 100% sensitivity, meaning detect every molecule that's there in the tissue. But we're getting there, we're on the way there, right? So now, obviously, this is a powerful data type, right? Because you've got gene expression and you've got spatial location. But the incredible thing is that, you know, that's this kind of technology, maybe a precursor to this, came out in 2015. Okay, and my colleague Kokha Chen published this paper in 2015. But he did not use the spatial information and the data when he was analyzing the data. And down even in 2021, and I'm sure if we look even in 2022, we'll find spatial omics papers where you run algorithms to say, cluster this data, cluster the cells by gene expression, and you throw away the spatial information in the clustering, which is crazy, right? So obviously, if you have information, you have to use it in your algorithm. So why is that, right? Because the space is so new. It's very, very new. And people are just working out what's the best way to incorporate spatial information in your data. And so we're taking a stab at that too, and we're proposing one way of doing that, but we feel is generalizable. I'll show you how to use that approach today for clustering, but I think it's a very general method. It can be used to solve a lot of spatial omics algorithms problems, not just clustering. Right? So can we incorporate spatial information cell type clustering? Of course, the answer is yes, we can, and people have done it. The question is how best to do it, and does it work well? So I'll talk today about two specific problems of incorporating spatial omics. They're both essentially clustering problems, okay? One is clustering cell types. So, and I'll describe what that means in a minute, and then the other is, meaning you're clustering in some gene expression space, okay? Each dot is a cell and your space is gene expression space, and you're clustering cell by similarity in that gene expression space. The other kind of clustering is you're clustering spatial locations. So you're grouping spatial locations together into a contiguous segment. So it's a segmentation problem. You're taking a 2D spatial omics image, segmenting it into connected spatial domains, and each connected domain is presumably a biologically meaningful unit, let's say a cortical layer, a layer in your brain, right? So these are two different clustering problems, one in expression space, one in, say, physical or geometric space. What are people doing here, okay? And these are the two problems I'll address today with this algorithm. So this is what the cell type clustering looks like. Here I'm just showing non-spatial single-cell RNA-seq data for illustration. There's, I think, like 300, 400,000 dots here. So it looks like blobs, but you have each dot is a cell. And this is some two-dimensional reduced dimensionality projection of gene expression space, okay? But it's a multi-dimensional, thousands of dimensions gene expression space. Cells that are near each other have the same cell type. If they're near each other in gene expression space, that means they're similar cells. And so they have the same cell type. So here the cells are colored by cell type. So these are T cells, these are NK cells, monocytes, platelets, etc. Right? And you can see here the really distinct islands are cell types that are very different. Within T cells, you have a continuum of gene expression. So these are naive T cells, these are effector T cells, naive CD4 T cells, naive CD8 T cells, and so on. So you have a continuum within a major cell type, and then you have discrete islands separating major cell types. So this is the cell type clustering problem in gene expression space, okay? Now, there's been not so many algorithms, but recently there's been quite a few algorithms coming out to do this spatially informed cell type clustering, okay? For various kinds of spatial omics data, what are the limitations? Still, there are relatively few algorithms out there, and they're not widely adopted. So mostly the people who publish the algorithm use it in their paper, but the broader field still uses non-spatial clustering. Perhaps one limitation is that they don't scale to large data sets, okay? What I mean, large data set, 600,000 cells in one single data set, one single tissue section. So that's what VisGen has just released publicly online, right? But all of these algorithms, they were tested on a few thousand cells. And as I'll show you, they don't scale even to 100,000 cells, most of them. And we have 600,000 cells today. Tomorrow we'll have millions of cells. Next year we'll have tens of millions of cells, right? So scalability is a huge issue in genomics, single cell genomics, and certainly in spatial genomics. Then the existing algorithms, they use some combination of deep learning, deep neural networks, autoencoders to transform your feature space, and Markov random fields, which is how does your expression vary across space, right? And they use either or both. But it's a question of how exactly do you represent the spatial information? What is your cost function and so on, right? And so it ends up having many spurious clusters, perhaps because the deep learning is overfitted or doesn't have enough data, or because in some cases, the assumptions of the Markov random field method are not valid. What do I mean by that? So let me quickly go back here. Okay, so the Markov random field methods, they assume that neighboring cells have the same cell type. Okay. But as you can see here, there's many regions in the brain where neighboring cells, even over here, this is relatively pure, right? You have different cell types interspersed. So that basic assumption of the Markov random field method is violated by most complex tissues. They still work okay, but you know, if you want optimal performance, you have to make assumptions that perfectly fit the data. So then that's cell type clustering, and then there's tissue domain segmentation. That's something like this, okay? These are the layers of, I think, the human dorsolateral prefrontal cortex. These are, each color is one cortical layer. This is manually annotated by experts, okay? Layers 1 to 6, and this is the white matter. So you want to segment your space into these contiguous biological structures. That's a tissue domain segmentation problem. So this is slightly older. So there's more algorithms in the space. Largely, again, I think the Markov random field paradigm, which actually is technically valid for this kind of data set. Because here in this data set, in one layer, developing brain, you have mostly homogeneous cell types, right? And that's why this data set has been used a lot for benchmarking these algorithms. But in general, that assumption is not valid. So again, I mentioned about the validity of the assumptions, and then some of the deep learning methods, they may overfit the data. Okay, so those are the limitations. How do we get around them? And so this is our stab at it. Algorithm is called building aggregates with a neighborhood kernel. I'll tell you what that means. And spatial yardstick, Banksy. There's a tradition in single cell to name algorithms after artists. So we chose a slightly more subversive artist, but still following tradition. I want to thank Vipul and Nigel. They are the two co-first authors of this, and the algorithm developers and benchmarkers primarily. And then my collaborators, Hui Kwan Lee and Kokhao Chen. So Kokhao is the first author of that 2015 Murfish paper. It was one of the seminal papers in this field for spatial omics technology. And now he's in Singapore, he's my colleague. So what does Banksy do? Just one very simple transformation of the data, and then everything else is conventional clustering, okay? What is that simple transformation? Banksy does feature augmentation, right? It says, what is a logical way to incorporate spatial information in the data? It just transforms the data. So this is your initial data matrix, okay? If I did not have spatial omics data, this is what my data matrix would look like. Each column is a cell, each row is a gene, okay? I have my genes versus cells expression matrix. I want to cluster my cells, so each column here is the expression vector of one cell, and I want to cluster the cells so that the cells near each other have similar gene expression vectors. That's my normal single cell clustering algorithm. So what Banksy does is it says, let's use the same machinery of conventional single cell clustering, except let's do feature augmentation. Let's double up the dimensionality of the feature space. So, and what's the biological logic here? It says each cell type has a characteristic transcriptome and a characteristic neighborhood, meaning because it has a characteristic distribution in gene in the tissue, right? And that means it has a characteristic neighborhood. Neighborhood means nearby cells. Okay. Now, this is the biological assumption, and it's sensible. What do we mean by neighborhood? How do we mathematically encode the neighborhood? So I've shown how we encode the cell's own transcriptome. It's just the gene expression vector of that cell. How do we encode the neighborhood? So the simplest thing you can do is just take the cell. So this is the cell's own transcriptome. Take the cell's immediate neighbors, k nearest neighbors in physical space in the tissue. Average their transcriptomes, average the transcriptomes of the k nearest neighbors, and append that to your original feature vector. So now you have a feature vector that's twice as long. That's all you're doing, right? So now you're encoding the cell by its own transcriptome and its neighboring transcriptome. So you're incorporating spatial information, but you're not making this assumption. You're not assuming that the nearby cells have the same transcriptome. You're allowing this vector to be different from this vector, right? That is much more realistic. You're also not saying that cells of the same type are necessarily near each other. Cells can be highly intermingled, right? So you can have, let's say you have this green cell type in this green neighborhood here. There may be a green neighborhood here too, and a green neighborhood here. That's okay, right? Each cell in a green neighborhood will have the green transcriptome, and its neighbors will also be green, right? So then cells of the same type will be clustered together, not just in this feature space, but also in this augmented feature space. And then there's a question of regularization or weighting. How much weight do you give to the cell's own transcriptome? How much weight do you give to the neighbor transcriptome? So then we just use a simple scale factor that's square root of lambda or square root of 1 minus lambda. So what this ends up being is that this should be d squared, okay, not d. So this is the Euclidean distance in expression space. So dE is the Euclidean distance of the cell's own transcriptome from another cell's transcriptome, and ds is the Euclidean distance of the cell's neighbor transcriptome from the other cell's neighbor transcriptome, okay? And so then you can see from these scale factors that the actual d squared in this augmented space is just a linear combination of the squared distances in the original space. When lambda is 0, this is 1, this is 0, and then you just have conventional single cell clustering because you're only doing the cell's own transcriptome distance. When lambda is 1, this term is 0, you only have the neighbor transcriptome, and that is a domain segmentation mode of Banksy. So we use a lambda of like 0.2 or something typically for cell type clustering, and a lambda of close to 1 for domain segmentation. So the beauty of this is you have one algorithm with just one simple free parameter that you can sensibly tune or intuitively tune. And that will give you either a cell type clustering algorithm or a domain segmentation algorithm. And after you do this, you don't need to do anything else. You can just take your established single cell RNA-seq clustering software and just plug in, you'll always do some dimensionality reduction, let's say using PCA. And then you just plug your PCA transformed version of this matrix into standard single cell clustering. And then it's highly scalable because existing single cell RNA-seq clustering algorithms are highly scalable, right? So this neighbor augmentation method strategy, it's biologically plausible, and we've used it for clustering. You can use it for any of the other, you can use it for, I don't know, trajectory analysis, you can use it for cell signaling analysis, you can use it for batch effect correction, really any algorithm in a single cell spatial omics space. And we hope to use it for all of those places. So how does it help? Okay, now let's say now the cell's own expression is a multidimensional vector. The cell's neighborhood expression is a multidimensional vector. Let's just simplify it by saying the cell's own expression is a one-dimensional vector. And the cell's neighborhood expression is also a one-dimensional vector, okay? So then each cell is a dot now in this two-dimensional space of own expression, neighborhood expression. You can see here we have these two cell types, blue and green. If you project them, if you discard the y-axis, you project them onto the x-axis, which is own expression, the blue cell type and the green cell type, they're separated, but they're somewhat intermingled also. So you have noise. These are ambiguous cells. You don't know what their cell type is. But because they have different neighborhoods, in this two-dimensional space, you can very cleanly separate them, right? And this cartoon is really the intuition of Banksy. And you'll see, I'll show you some examples where this exactly works, where in the own expression space, it's fuzzy, with neighborhood expressions really crisp. And so the tagline is, a cell is known by the company it keeps, which I thought was very original, but it turns out that in natural language processing, people came up with this, I think decades ago, where they said a word is known by the company it keeps. So some of you may know that natural language processing, words are encoded by the frequency of their neighboring words in the text. And that tells you something about the meaning of that word, right? And so that's a very nice analogy, which we found out only afterwards, after we devised the algorithm, that people in natural language processing have been using this strategy already for a long time. And but it works for cells also. So I've explained this part. So but I'll just reiterate briefly. As you adjust lambda, you swing Banksy from either a cell type clustering method or a domain segmentation method. Okay? So which is either in one extreme of clustering on the x-axis and other extreme of clustering on the y-axis. Now, does the method work? Now, any algorithm can sound plausible initially. You really have to benchmark it, and we spent a lot of time benchmarking it on many, many different data types, not just one data type, not just one data set. And as you'll see, Banksy doesn't have too many free parameters. We didn't have a lot of room to fiddle. Essentially one free parameter.
How do you define neighborhood? Is it spatially defined by a radius around the cell, or is it
exactly.
K neighbors?
K nearest neighbors.
K.
Yeah. By Euclidean distance in the tissue space. So here's a Murfish data set from the same lab that Prabhakar graduated from, Shyam's lab. It's a Murfish spatial omics data set, mouse brain again. You can see here there's one tissue structure here and a few more here. Mouse hypothalamus. Okay. Now, if you look at conventional clustering, non-spatial clustering, if you ignore space, okay, all these yellow cells all show up in the same cluster. But you can see here that there's one structure here, one structure here. Here the yellow cells are dense, here yellow cells are sparse. Banksy actually separates them out into two different clusters, okay, very, very crisply, as two different subtypes. They're both oligodendrocytes, they're both mature oligodendrocytes, but they're two different subtypes. Is that correct, right? So here's the first thing we do to make sense of our results is we look at the gene expression matrix. So here are some marker genes of these two cell subtypes. Marker genes means genes that have differential expression in those two cell types. This is cell type 1, this is cell type 2, each column is a cell, each row is a marker gene. This is the cell's own expression. This is cell's neighbor expression of the same marker genes. You can see here that in the neighbor expression space, these two cell types are very crisply separated, which kind of makes sense. The cell type composition of these two neighborhoods is different, right? But maybe that's an artifact. Maybe the cell's own expression is exactly the same, in which case we'll say Banksy is making a mistake. It's fooled by the differences in the neighborhood to assume that the cell itself is different, right? But actually, if you look at the cell itself, you see starkly different expression. Actually, it's this color scheme, okay? It's statistically, it's different, but it's not hugely different, okay? These markers and these markers, you see a statistically significant difference in expression. So that means the cell's own expression is also different in these two neighborhoods, but it's a subtle difference. It's not as strong as this difference. And that's why conventional clustering that ignores spatial position will not find the subtle distinction in these two cell types. With spatial position, you can find it. And that's the power of spatial omics, and that's the power of this neighbor augmentation. Visually, it's so easy to see, right? Why neighbor augmentation helps to separate these cell types. And we have many more examples here. I'm just zooming in on this one, because if I remember correctly, this is a distinction that the other algorithms could not catch. The other spatial omics clustering algorithms could not catch this distinction. So here's same mouse hypothalamus omics data set, another pair of cell types. These are excitatory neurons. These are two different sections of this mouse brain. And you can see again, non-spatial clustering says they're all the same subtype of excitatory neurons. Banksy says that they are two different subtypes with two different spatial locations. Here, again, neighbor transcriptome says they are different cell types. Own transcriptome again says they are different cell types. So this is the validation. Okay, you always have to eyeball this and say is it correct or not. So beautiful. It's giving you spatially, it's giving you meaningful results. I don't have the result here, but we validated all of these by looking at single cell RNA-seq also, right? You want some external validation. Now, in single cell RNA-seq, these two clusters don't separate out cleanly, but if with your conventional clustering, right? But if you tell the single cell RNA-seq clustering algorithm, only use these genes. Now we know which genes to look at. We know the feature selection. If you only use these genes, then these two cell subtypes do separate out even in single cell RNA-seq. Right? So it is independently possible to validate this. It's just that it's hard to know that these are the right genes to cluster when we know it because of the spatial data.
Can you clarify? So those genes that cluster here, were they previously known to be potentially associated with different subtypes?
They were not known, as far as we know. We just found these two clusters, and then we just did a hypothesis test with differential expression analysis between those two clusters. And that's how we've identified
the
these
diagnostic gene set that you can then use in the single cell.
Exactly. We did the gene differential expression analysis between these two clusters to find these genes. Then we looked in single cell data. But we didn't know about these genes before. But I'm sure if we dig deep enough in the literature, someone would have said something about some of these genes. Okay, another data type, Slide-seq. So Slide-seq is also a spatial omics, but you have beads. Okay, each red circle is a bead as used to capture gene expression in this spatial region. So you can see here that neighboring cells, their expression can be confounded in one bead. Okay, in one spot. So it's hard to cluster this kind of data. So again, Banksy, so this is non-spatial clustering of this brain section. You can see here that each cluster is like slightly messed up, okay, in the non-spatial clustering. So there was a very nice paper presenting this RCTD algorithm for deconvolving these mixed up bead transcriptomes. And then you get a cleaner representation, you get cleaner clustering of each individual cell type. But the cost is you need to know in advance, you need a reference data set of the transcriptomes of these purified cell types. And you need that independently from single cell RNA-seq data. Only then can you use this deconvolution method. Banksy does not need deconvolution. It just takes the spatial omics data, no external data, and very crisply finds these cell types because it uses the spatial information intelligently. And yeah, so that's a big advantage, just much more convenient, right, for the Slide-seq data analysis. More examples. This is another mouse hippocampus brain data set. So you can see here we're comparing Banksy to non-spatial clustering and various other algorithms for spatial clustering. So these are 3 other algorithms for spatial clustering. And you can see here that, so the colors indicate the cell types. These cell types are all known cell types now. These are not new discoveries. They're known cell types. They're kind of ground truth positive control. But the existing algorithms, they cannot cleanly separate out these known cell types in all cases. Okay, so some cell types are merged together here that are actually distinct. Over here, there's a lot of merging. And then over here also there's over merging where, you know, there should be, it should not be so dense over here. And I think this cell type was not cleanly identified in this algorithm. Yeah, many more. If you look at this, is preprinters on bioRxiv, many more such examples. I want to switch gears to domain segmentation, okay, where identifying tissue domains. The simplest thing is find, this has not been done before in spatial omics data, but you can imagine, say, in a brain organoid, right? You have some misdifferentiated regions. You have a necrotic core where you're culturing the organoid and the nutrients are not getting into the core of the organoid, so the cells start dying in the core of the organoid. So these are all spatially contiguous domains that you want to segment, and then you want to kick them out. So this is a QC step. So we use Banksy for this kind of QC, and very nicely found these misdifferentiated zones, and also these the necrotic zone much more cleanly. If you do non-spatial clustering, or you know, it doesn't work so well. Markov random field still works reasonably well. Okay, but this is, I think, a novel application of this kind of spatial clustering for this data QC. And my collaborator, Jinyue Liu, actually used this in her work for brain organoid morphish data. And then this is the famous benchmark data set I mentioned. This is dorsolateral prefrontal cortex, manually annotated ground truth, within quotes, ground truth. Layers, which algorithm identifies them most accurately. This is slightly older. We benchmarked more algorithms now, but the take-home message is the same. The y-axis is the similarity of the algorithms clustering to the ground truth, the manual ground truth. You can see here that Banksy just has, on average, the highest similarity to the manual ground truth of all the algorithms. And it's the same Banksy algorithm, we just changed lambda, we just increased lambda. Okay. So whereas the others are all algorithms that are specifically designed for tissue domain segmentation. And it's not obvious why Banksy does better at first glance, because it's such a simple method. You're just smoothing, it's just a kernel smoothing of the neighborhood, right? And I think it's simply because these other methods, either they make invalid assumptions or they overfit the data. It should not be possible to outdo a simple smoothing algorithm, right? So I think there's a lot more room for Banksy to grow and become even better. But at least it avoids some of the pitfalls of the existing methods. So lastly, scalability. X-axis is the number of cells, and we tested Banksy on up to 1.5 million cells. You can see it has the same runtime as non-spatial clustering. The existing methods, some deep learning, Markov random fields, they simply crash or fail to return a result very fast, a very small number of cells. And that's not usable on large data sets. So to summarize, Banksy has a unique biologically motivated feature augmentation strategy. Is the first algorithm to combine 2 different problems together, cell type clustering and tissue domain segmentation. And we can apply to many data types, and I think other algorithmic problems also. And perhaps because it's simple and intuitive and biologically sensible, it does better on all the benchmarks we tried. And it finds new cell types actually, which are interesting to explore. And it's very simple, very robust, orders magnitude faster than other methods. I've thanked Vipul, Nigel, my major colleagues, It was funded by A-Star, and also we had a collaboration with Veranome, which is one of the companies in this space. Thank you.
So thank you, very nice talk. So have you tried to analyze cell-cell communication, taking Banksy into account?
That's so one of my postdocs wants to do that, but the algorithm hasn't been done.
You haven't done it yet, so you don't know whether you find different types of communication for the populations that are very coherent and the ones that are mixed up.
We don't know yet, but it's a very good idea, I think, to look at. I'm sorry, I don't know who raised that first.
Yeah, thanks a lot for the nice talk. So this seems very similar to a graph clustering method. So did you also kind of compare it with like?
Yeah, we use graph clustering actually.
Yeah, so then I think my question is more on that. Did you also find neighbors of neighbors as an interesting feature to add in, or like?
We have not gotten so fancy yet. So this is the first time.
Yeah, I was just wondering from a biologics perspective, if you say you have identified novel cells, new cell types from the clustering, have you done this on some sort of model organism or so where you can easily genetically manipulate those and actually biologically confirm there's an underlying biology and it's not just a little bit of variation in the expression patterns that you detected because of them?
Great question. I think the first thing we need to do is fact purify those cells, those subtypes, and say, do they have any functional difference? We're just functionalizing these things in vitro. We've cultured neuronal cells in vitro. They lose their function. They lose their in vivo properties. It's very hard, especially when you have such subtly different subtypes. So I think something in vivo, like what you're saying, maybe genetically perturb them, is more feasible. That will take 3 or 4 years. So sorry.
Sir, no. It's a follow-up question on the earlier question and something on your question related to the choice of K, the nearest neighbors. How many neighbors do you choose? Is it a free parameter, something you set, and how does it affect the results it sends?
I think it's very robust. So we use somewhere between K equals 30 and K equals 300. There's almost no difference in the result. I think we use K equal to 50 as a default.
You said lambda when you know it's not true.
Well, lambda is kind of like the resolution parameter in single-cell RNA clustering. And for this kind of clustering algorithm, you never have ground truth, right? So you adjust your parameter and you eyeball it. You look at the markers. You say, does it make sense? Does it not make sense? That's true for any such clustering. But on average, what we found by trial and error is that lambda equals 0.2 works for cell type clustering almost everything. So we just keep it at lambda equals 0.2 generally.
Because whenever you have ground truth, like in this one of the examples you show, you know what would be a proper separation, right? But if you don't know what is the proper separation, you can create more or less separation. Do you have any suggestions how to tune that in the case where we don't really know what to expect?
To be honest, it's just empiricism. For all the data sets I've shown here, after we analyze them, reading the literature, looking at single-cell RNA-seq, etc., now we have some sense for the ground truth. It's not bulletproof, but some sense. And lambda equals 0.2, 0.3 seems to work reproducibly across different data types, different data sets, and so on. That's all. It's still empiricism. I don't think there's a theoretical way to say lambda equals 0.2 is the best way to do it. And that's why it is a free parameter.
Two things. First, Mr. Lambda, do you think it's accurate to make it a global parameter, or do you think it's more like a local parameter that in some neighborhoods you on one and in other neighborhoods you on two is better?
Yeah. Exactly, yeah. So that makes complete sense. And so that would be like a more fancy version of the other.
And the other thing, segmentation is always an issue in those fish-based assays. Slide-seq 2, it's by design, basically. So you have some contamination from the cell chef to the neighbors and the opposite way around. You try to reduce this contamination, or you think that it doesn't matter?
So we've segmented Slide-seq data and Visium data Yeah, they all have contamination from neighboring cells. I suspect that even when you have high resolution data, like subcellular resolution data, you still want to segment, and you will still need this method because you'll still need information from it.
Excuse me. With the choice of say Euclidean distance and also like how you define the neighborhood, would that like affect the performance of your method, for instance, when you use KNN? Because I feel like the spatial distribution of the cell density will also like heavily affect how it performs based on that.
Absolutely. So, for example, there's some very thin layers, there's some very fat layers, round layers, elongated layers. So all of that. So again, all of that is something we didn't take into account. But I think a more sophisticated method would probably maybe do some EM approach to say how many neighbors to choose for this region of the tissue, how many spatial neighbors, how many expression neighbors, and so on. So I think it's possible to do iterative optimization and localization of that. But right now it's kind of one size fits all. And that makes it very fast.
Yeah, so if I'm not mistaken, with the cluster, all of the patterns that you were looking at were single. So trying it on other such like Visium that gives me more average number of cells.
Yeah, so this is Visium.
No, not the segmentation, the clustering.
Clustering, we have not benchmarked it on Visium. Yeah. So Visium, at least at the current resolution, 100 microns, it's just hard to identify cell types, right? So only domain segmentation makes sense. Visium HD, I think we can start to look at cell.
But the clustering requires.
Yeah. Or almost single cell, like Slide-seq is almost single cell.
Okay, let's thank the speaker.
Thank you.
Now on a break till 2:30.